Alright guys, so this is going to be round 2 of plant transport and nutrition. So I've already made this video once, but apparently with my new computers I did not have the audio on. So we are going to try it again and see how it goes. So this particular lesson is going to be all about plant transport and nutrition. And it's actually a pretty short lesson, and a lot of what we're going to cover is a review of things that you should already be familiar with. And so we're just going to start off with just some basic, very general transport mechanisms. And this actually goes back all the way to first semester when we covered cells and the cell membrane. And so we're going to have both passive and active transport working in the movement of substances in plants. And so just as a quick review, remember that passive transport, anytime you're moving substances from high concentration to low concentration, that does not require energy. And this is a natural movement. Molecules naturally want to move from where there are more of them to where there are less of them. So this does not require any input of energy. This is just a very natural downhill movement. Now, active transport, on the other hand, is the opposite. We're going to be moving things from low to high. And so anytime you're trying to move molecules where they are already highly concentrated, you're going to have to give them a push. You're going to have to input energy to do that. And so this typically requires proteins that we call pumps. One example is a proton pump. We actually took a look at this when we did cellular respiration. We know that we can create these concentration gradients, which give us potential energy. And so in plant transport, we're going to see a combination of things moving from high to low, very passively with no energy, but we're also going to see some things moving low to high and being pumped. And then co-transport is when we kind of combine the two together. So a lot of times what will happen is we'll use a pump to create that potential energy. We'll concentrate something on one side of the membrane, and then as it flows downhill, it's going to actually provide energy to pump something uphill. And so as one molecule is moving down, that powers another molecule to move up. And so the two molecules are moving together, and so we call that co-transport. Now, osmosis is a super important one because osmosis is the diffusion of water. This is a passive type of transport. Water is going to move from wherever it is higher or wherever it has a higher water potential to where there is less water or a lower water potential. The more solute something has, the lower the water potential it has. And remember that solute sucks, okay? Water is always going to move to an area that has more solute. And water can and will move because it passes through these proteins called aquaporins. So remember, the cell membrane is a phospholipid bilayer, and it has those hydrophobic tails in the middle. And so water can't really pass across the phospholipid bilayer, but it can move through these proteins called aquaporins. So these are going to allow for that water passage. And so anytime you do have a water imbalance, water is going to move, okay? Water is going to move through those aquaporins. And then we also have a transport mechanism known as bulk flow. We're going to see that in the xylem and the phloem. And this is a large-scale movement due to pressure changes. If you've ever created a siphon, like siphoning gas, or I've seen my sister take water out of an aquarium by doing this, but you take an empty tube, you suck on one end of it, it creates this pressure, and once it starts flowing, the same kind of concept works with like pool vacuums and things like that. But when you have a change in pressure, it can create a flow. So high pressure on one end, low pressure on the other end, and those substances, those fluids will just flow right on through that tube. So this right here is a picture of an aquaporin. Okay, these are little tiny water molecules, and you can see that this little channel is designed for these water molecules to pass. And so that's why water will move anytime it has an opportunity. And then over here is a picture of xylem and phloem. So we talked about vascular tissue in our first plant lesson. In xylem, it's always carrying water upward. In phloem, it's going to be carrying sugar and other materials from source to sink. And that's what we're going to take a look at here in just a moment. So in addition to things moving across the membrane by passive and active transport, we also have some lateral transport. And this is how it's going to move from cell to cell. So if it could be transmembrane, which is where you have cell walls and cell membranes, and it's literally just moving across, all the way across from one cell to another. Symplastic is where it moves through plasmodesmata. So remember that in that rigid cell wall are little tiny channels called plasmodesmata, and that allows for that cytoplasm to be continuous. So these plant cells are not isolated by their cell wall. They can actually still communicate and pass substances through these little tiny channels, and the cytoplasm is continuous all throughout that plant from cell to cell. Or it could be apoplastic, which is where it's traveling through the extracellular spaces. And so I have a picture here. So here's my apoplastic. You can see that it's staying within the outside and the cell wall. Here's the symplastic. It's passing through that plasmodesmata, which that's a little bit blown up. They aren't quite that large. If it were transmembrane, it would be moving this way. It would be moving through the cell membrane, through the cell wall, into the neighboring cell. All right, so now we're going to take a look at the specifics of water and sugar transport. We're going to start with water, and water is going to be moving upward, okay? For water, it's pretty much always an upward movement, and that is happening in the xylem, okay? So this whole slide is what's happening in the xylem. Hate this highlighter. And so hopefully you know this, but water is going to be absorbed at the roots. Water really can't enter the plant through the leaves and the stems, because remember that shoot system typically has a cuticle on it, that waxy layer, and wax and water don't mix. And so the water is always going to be coming in, just about. There may be a few exceptions, but pretty much water is going to be coming in at the roots. And so the water has to travel upward. So we're always going to have an upward movement of water in the xylem, and this is defying gravity. If you think about a tall tree, the water has to get all the way to the top. And so we're going to have some serious forces at work in order to be able to make that water move. And so we're actually going to have three forces. I like to sum it up with a push, a pull, and a climb. And so we're going to take a look at three mechanisms that are going to help to keep this water moving up in the plant at all times in the xylem. Okay, so we're going to have a push, a pull, and a climb. So we're going to start with the push, and the push is known as root pressure. And so what happens is we have substances, we have nutrients, we have minerals, we have ions. They are going to get pumped into the root by active transport. Okay, so several minerals and nutrients will be pumped into the root. Now, we know already that solute sucks. And so what happens is when these minerals and ions and nutrients get pumped, they're solutes. And so as soon as they get pumped into the roots, that is going to pull water in, because that lowers the water potential in the root. So now water is passively going to flow in by osmosis. And so that inward push of water into the root is known as root pressure. And this is going to help push those fluids up the xylem. This is also what causes the dew to come out of the grass in the morning. So that's our first force, okay? Now, our second force is going to be a pull, and that is going to be created by transpiration. And so transpiration is the water loss from the leaves. So those little tiny stomata, those holes on the underside of leaves, they are intended for gas exchange. But when the stomata are open, water can leave the plant. Okay, so this is actually a big checks and balances that the plants have to deal with. And so this is what your virtual lab is going to be on, is transpiration. And so what happens is that the water evaporates. And as the water evaporates from those leaves, it creates this negative pressure. As the water evaporates, it kind of pulls on the water molecules behind it and helps pull that water upward. Okay? So we have what's called transpirational pull. All right, so our push was root pressure, our pull is due to transpiration, and then our third is going to be a climb, and that is going to be capillary action. So back when we covered properties of water, we took a look at cohesion and adhesion. Now water is sticky. Okay, that's what these words are telling us. Water is sticky. Cohesion is when water sticks to itself, like how it can bead up and form a bubble, or you can overfill a cup. And adhesion is when water sticks to other things. And so the combination of the stickiness of water, of cohesion and adhesion, water will literally climb tubes, okay? And this is what we call capillary action. Due to stickiness, water will climb up a tube. So you put celery in water, that water will climb up the little tubes of the celery. You don't have to do anything. So between the three, we have root pressure, where water is flowing into the root, pushing upward. We have transpiration, where water is evaporating from the leaves, pulling on the water molecules behind them. And then we have capillary action, which is going to be a natural climbing due to the stickiness of water. So this is what always allows water to move upward in the xylem of a plant. All right, so here you have the picture. This is root pressure. So you have ions and minerals that get pumped into the root, and then water is naturally going to flow in after it. So we have root pressure here, we have a natural climb through those tubes, and then we have a pull from transpiration at the top. Now sugar transport is going to be a little bit different because with water it's always moving upward, but with sugar it's not necessarily always in the same direction. So we kind of summarize the movement of phloem in phloem as saying source to sink. So your sugars are going to move from source to sink. The source is where they're made, which is typically the leaves. Those are your main photosynthetic centers. And then the sink is where they're stored. Now that's going to vary depending on the plant. Okay, sometimes they could be stored in a fruit, sometimes they could be stored in a root, sometimes they could be stored in a modified stem like a potato or a tuber. It could be stored in a bulb, which is also kind of a modified leaf. And so it really just depends on the type of plant that you have. But somewhere those sugars are going to be stored in the form of starch for that particular plant. So it's not necessarily always down. It really just depends on where your sink is. Okay, so in water, the movement is upward, but in phloem, the sugars are moving from source to sink. And this is going to happen by that bulk flow mechanism that we mentioned earlier. So it starts when the sugars are loaded into the sieve tubes, okay? So the sieve tubes are some specialized components, some specialized cells in our phloem. And so what happens is that sugar is going to be actively transported into the sieve tubes. Now, as we saw earlier, solute sucks, and so once that sugar gets in, it's going to cause water to naturally flow in after it. And so this loading is going to create a positive pressure, okay, as sugars are pumped and water flows in after. Now, once we get to the sink, what's going to happen is the opposite. Those sugars are going to be loaded into the sink, and then water is going to flow out, and that is going to actually create a negative pressure. And so here we have a positive pressure, here we have a negative pressure, and that allows for that bulk flow. Okay, so a lot of times you've heard of maybe sap flowing, and it really does flow in the phloem as the sugars are loaded and unloaded. All right, so the last part of our notes, we're just going to take a look at some plant nutrition and some relationships that plants can have. A lot of this is a review of things that you probably already know. So we're going to start with the rhizosphere. And we've actually seen this prefix before when we saw mycorrhiza. This R-H-I-Z, rhiz or rhiza, it's actually in reference to root. Okay, so your rhizosphere is essentially your roots and all of that soil material that is around it. Okay, very important for that plant. There's lots of mutualistic bacteria. This just means soil bacteria. And so these relationships are definitely win-win. The plants are providing food and compounds, and then these bacteria are providing things like nitrogen fixation. They may be providing chemicals like hormones or other nutrients. And so the rhizosphere is just kind of that, you know, if you pull a plant up by the roots, it kind of comes up with soil intact. All of that is going to be your rhizosphere. Now within that rhizosphere, you may have these rhizobium or these nitrogen-fixing bacteria. We learned about the importance of nitrogen fixation back with the nitrogen cycle. So remember that nitrogen is very abundant in the air, but living things cannot use it in that form. Okay, plants cannot use it in that form. We really need either environmental changes like lightning or forest fires, something like that, or we need these bacteria. And so these bacteria specifically live in legumes, which are bean plants. And so what they do is they have the ability to take this nitrogen gas and turn it into usable nitrogen compounds. So then the plants are able to use it, and then it can pass through the food chains. And so pretty much all living things are going to be dependent upon these rhizobium bacteria to fix that nitrogen, because most living things cannot do it for themselves. We also have mycorrhiza, and so this is the term that we've seen before. Remember, myco is fungi, rhiza is root. So this is that mutualistic relationship where the plant provides food, and then the fungi are very absorptive. Remember, they have those hyphae, and so they absorb lots of water and nutrients and share those with the plant roots. Some other relationships that you might see, we do have parasitic plants, which means they are stealing nutrients. And so what they do, like this mistletoe up here in the tree, they, instead of like having their own root system, what they do is they tap into the vascular system of another plant. And so they're literally stealing water and stealing sugars and stealing nutrients from the xylem and phloem. And this does hurt the host plant. If you get too much mistletoe on a tree, it will kill it. Now, sometimes your parasitic plant may be photosynthetic and be able to make its own food, but anytime it is stealing anything, it is going to be harmful to the host plant. Now, epiphytes kind of look like parasites, but they are not. Okay, here's a picture over here of an orchid, but they are plants that grow on other plants. And they do this to get closer to the sunlight. So it's pretty common to see epiphytes in the rainforest, where you have the canopy that shades the lower levels. And so plants need sunlight to grow. And so what they'll do is they'll grow on another taller tree, but they have their own means of obtaining water and nutrients. A lot of times they have aerial roots. And so they're just literally using the tree for a growing medium. They're not stealing anything. They're not hurting the tree. So epiphytes are actually an example of a commensalistic relationship. And then you have carnivorous plants. Now, a lot of times they are photosynthetic. They are still making their own food, but the reason they digest little tiny organisms is because the soils don't have good nutrients. So there's going to be other things that they need beside the sugars that they make, some minerals and vitamins and ions and things like that. And so they are able to kind of trap and digest little insects to get those nutrients that are lacking in their soils. And that is going to wrap up our lesson on plant transport and nutrition. Like I said, you have a Transpiration virtual lab that you're going to be doing to kind of practice some of these concepts. You also have a study guide that I would encourage you to take a look at. It is some excellent practice. Here soon, we're going to be doing our last plant lesson on plant reproduction. And I just want to let you know that you're going to have one big quiz. Your quiz is going to be over all things plants, and so it'll be coming probably either later in the week or early next week, but just plan on taking a quiz over all things plant. So I hope you guys have a great day. I hope you had a good break. Just a few more weeks and we'll be wrapping up DC Bio. I hope to see you soon. Bye, y'all.